Hello and welcome to all my dear students. So, today I am here with the new lecture of the chapter Principles of Inheritance and Variation. I hope that you all have studied the past lectures well and have understood each and everything. Today, beta, we are going to talk about mutations. Now, if you must have seen the last lecture, then you must have seen that mutation we have already started, and one type of mutation, that is gene mutation, we have also already discussed. But to those students who are watching this lecture but have not gone through the past lecture, for them we are going to start from the mutation from the beginning. So today we are going to basically discuss about mutation. Let me give you a little bit hint about what we have discussed in our last lecture. So what is mutation, beta? Mutations are basically sudden changes which occurs in the genotype and phenotype of an organism. Now, mutation term was given by Hugo de Vries while he was working on Oenothera lamarckiana, commonly called as evening primrose. Beta, mutations can be inheritable or cannot be inheritable. That means mutation can be passed from the parents to the offspring, or they may also do not get passed from the parents to the offspring. Right? Now beta, mutations can be of three types. Gene mutation that we have already discussed. Today we are going to discuss about chromosomal mutation and genomatic mutation. Along with this, we are also going to discuss the agents causing mutation, that is mutagen. Now, first let me just give you a quick revision of what is gene mutation. So basically beta, gene mutation means when a mutation is occurring in a specific nucleotide of a gene located on a chromosome, right? There you have seen two types of mutation under the heading frame shift and substitution mutation. Today, we are going to continue with the second type of mutation, that is chromosomal. Now beta, it is slightly different from gene mutation. How? In chromosomal mutation, actually there is a change in the chromosomal arrangement. The number of chromosome will remain same, but within the chromosome, when the mutation is occurring in more than one gene. In gene mutation, change was occurring in only one gene within the nucleotides of one gene. But in case of chromosomal mutation, when number of genes are affected on the same chromosome without changing their number, is known as chromosomal mutation. So what is chromosomal mutation, beta? When the change is occurring in more than one gene of a chromosome. More than one gene of a chromosome without changing its number. Without changing the number, the total number of chromosome. Without changing the total number of chromosome. Right? Under chromosomal mutation, we have four types: deletion, addition, translocation, and inversion. So basically, you have to study 4 to 5 types of chromosomal mutation. First is deletion. Deletion means beta, when a segment of chromosome consisting of more than one gene gets deleted. So what is deletion mutation? Deletion chromosomal mutation means when a part of chromosome, maybe upper arm or lower arm of the chromosome, including more than one gene, when a part of chromosome consisting of more than one gene gets deleted. So this is known as deletion chromosomal mutation. Like for example, if initial, the chromosome is this. Suppose it is represented by number of genes. So this is the centromere of the chromosome, which is going to keep the two arms, upper and lower arm together. It is having different genes like A, B, C, D, E, F, G, H, I, J. So these are the different genes located on the chromosome. Now, any part, maybe upper arm of the chromosome or the lower arm of the chromosome, if gets deleted. Imagine if this portion of the chromosome gets deleted, mutation occur, and now the chromosome is only represented by gene A, B, C, D, E, F, G. Now, HIJ portion of the chromosome got deleted. Now, what will happen? These three genes will be absent in the progeny, because of which the work, suppose these three genes are producing a protein controlling mental ability. Now, the person who is lacking these three genes will not produce the protein controlling mental ability. Such a person will become mentally retarded. So, mutation is causing change in the genotype as well as change in the phenotype of an organism. Now, the organism will be mentally retarded. So, this is one of the type of chromosomal mutation, that is deletion. Second is duplication mutation. Duplication chromosomal mutation means when same segment of the chromosome gets repeated, duplicate. Correct? So when the same segment of chromosome gets duplicated. Right? When the same segment of chromosome gets duplicated, then it is known as duplication mutation. Like for example, see, this was the chromosome having different genes like A, B, C, D, E, F, G, H, I, J. Now, if duplication mutation is happening, then any segment of the chromosome will get doubled. Like for example, if GHI gets doubled, so GHI, again GHI will occur, and then J. Here A, B, C, D, E, F gene. So upper arm is normal, but the lower arm of the chromosome doubles the number of gene, duplicates the number of gene. So as a result, amount of protein will get increased, the amount of protein produced, thus in the progeny will get increased, because of which, because of the increase in the total number of amount of gene, amount of protein, again some trouble, some change in the phenotype of an organism will be reported. Clear? Understood? So this is known as duplication mutation. Third type of chromosomal mutation is translocation. Translocation, beta, means transfer of a gene segment or transfer of a chromosomal segment from one to another non-homologous chromosome. Now, normally you know that there is transfer of genetic material, but between homologous chromosome, which is actually called as crossing over. Now, suppose the same exchange, suppose the same transfer of genetic material takes place, but between non-homologous chromosome, then that is actually a mutation. That is called as illegitimate crossing over. Illegitimate means not accepted, wrong crossing over. which will cause number of problems. Like for example, there is one type of cancer caused due to illegitimate crossing over. If a part of chromosome 22 gets shifted to chromosome 9, if crossing over takes place between chromosome 22 and chromosome 9, now these two are non-homologous, but if still exchange of genetic material takes place between them, a part of chromosome 22 goes to chromosome 9, then this is known as illegitimate crossing over, and it causes a type of cancer called as Philadelphia cancer. Correct? Understood? So what is translocation? Transfer of a chromosomal segment between non-homologous chromosome. Between non-homologous chromosome is known as translocation. It is also called as beta illegitimate crossing over. It is also known as illegitimate crossing over. Like for example, a long arm of chromosome 22 gets shifted to chromosome number 9. So yes, when a part of chromosome 22 gets shifted to chromosome 9, this is actually illegitimate crossing over between the non-homologous chromosome. So it is going to cause some or the other change in the phenotype, and it causes a type of cancer called as Philadelphia cancer or Philadelphia leukemia. Understood the three types of chromosomal mutation? Chalo. Now, the fourth type of chromosomal mutation is inversion. Inversion means beta rotation of the genes located on chromosome at 180 degree. So what is inversion? When the genes located on chromosome beta, when the genes located on chromosome moves at 180 degree or rotates, rather than move, write the term rotates at 180 degree like this. There are two types of inversion beta. Paracentric inversion, I'll explain, but first let me write it. Inversion without involving centromere. Without involving centromere. Whereas pericentric is type of inversion which is involving centromere. I'll show both with the help of diagram. Inversion involving centromere. So now let me show you. First, I am going to show what is paracentric type of inversion. Suppose this is my normal chromosome having genes A, B, C, D towards the upper arm of the chromosome and E, F, G, H towards the lower arm. Now see beta, this is the lower arm and this is the upper arm. If paracentric inversion will take place, then rotation of genes will occur without involving centromere. That means the new DCBA. So this is known as paracentric inversion. Can you see? The gene arrangement have rotated at 180 degree. Initially, the genes on the upper arm were arranged as A, B, C, D. Now, due to paracentric mutation, now what is the arrangement of the gene? D, C, B, A. The genes have rotated at 180 degree, but they have not included centromere. The things are happening without including centromere. Now, if the same type of inversion has to take place via pericentric, so beta, what will happen in case of pericentric? Okay, this is the original condition. A, B, C, D, E, F, G, H. This is the original condition, arrangement of the gene. Now suppose if pericentric type of inversion chromosomal mutation will take place, so beta, in that case, the arrangement will become A, B, E, F, then centromere, D, C, G, H. Now see, initially arrangement was C, D, E, F. Now arrangement have become E, F, D, C. So arrangement of gene has rotated at 180 degree involving centromere. Rotation is occurring around centromere. So that is known as pericentric. If rotation is taking place but not involving centromere, then that is known as paracentric type of inversion. Clear? So this is your chromosomal type of mutation where without changing the total number of chromosome, actually the arrangement of gene, the arrangement of chromosomal segments are getting altered. They may show deletion, they may show duplication, they may show translocation or inversion. Understood each and every one of you? Okay, very good. Now, the third type is genomatic mutation. The third type of mutation is genomatic. देखो, gene में क्या किया बच्चों? In gene mutation, there was a change in the nucleotide sequence of one gene. In chromosomal mutation, without changing the number of chromosome, we have actually altered many genes located on the same chromosome. Now, third type genomatic means changing the number of chromosome. Now, maybe whole chromosome will get deleted or added. So like this, total number of chromosome in an organism will change in case of genomatic mutation. Now, that change can be either in multiples of basic set or not in multiples of basic set. Clear? So what is genomatic mutation, bachcho? When overall the number of chromosomes are getting altered. The number of chromosomes are getting altered, maybe in multiples of basic set or not in multiples of basic set. So इसका क्या मतलब है? What does this line means? Multiples of basic set. चलो. So in order to explain this, I will explain first the two types of genomatic mutation, Aneuploidy and Euploidy. So genomatic mutation can be broadly divided into two categories, that is Aneuploidy and Euploidy. Aneuploidy means beta when the number of chromosomes are changing but not in multiples of basic set. When the number of chromosomes are changing but not in multiples of basic set, but not in multiples of basic set. क्या मतलब है इसका? Suppose मान लो बेटा there's a diploid cell which is having 8 chromosome. That, or to explain in a better way, let me take number of chromosome even lesser. Suppose there is a diploid cell having only 4 chromosome. That means 2 pair of chromosome. 4 means 2 pair. Now, this is 1 pair and this is another pair. So, this is a diploid cell which is having total 4 chromosome represented by 2 pair. Ab, if aneuploidy beta type of mutation has to take place, then now over here basic set is actually 2. The basic set for this cell, basic set is represented by X, right? So X over here is 2. Now aneuploidy means change in the chromosome number but not in multiples of basic set. That means either addition of chromosome is taking place or deletion of chromosome is taking place but not in multiples of 2. That means either 1 chromosome is getting added, 3 is getting added, 2, matlab not in proper multiples of the basic set. See how? Up suppose addition of chromosome has to take place. So if in multiples of basic set addition has to take place, then this is the way how it should have been done. This is one complete set, X2. This is another complete set, another 2. This is another complete set. So if like this addition or deletion of chromosome is taking place in multiples of basic set, then it is known as euploidy. And if it is not taking place in multiples of basic set, then it is known as aneuploidy. Right? Now let's see the types of aneuploidy mutation. First two are the types of deletion aneuploidy under the name monosomy and nullisomy. First of all, the best example of monosomy which is there in your syllabus is Turner syndrome. But before I tell you something about Turner syndrome, let me tell you what is monosomy type of deletion aneuploidy. Okay, suppose this is a diploid cell having 8 chromosome. That means how many pair? 4 pair. That means basic set will become 4. Right? So see, this is 1 set, 2, 3, 4. Correct? Or let me draw it in a proper way more. This is a cell, right? Having how many pairs of chromosome? 4. 1, 2, 3, and now this is 4. Now beta check this. This is representing one complete set. And this is representing another complete set. In monosomy, what happens? One of the chromosome of a pair gets deleted. That means condition will become 2n minus 1. So what happens in case of monosomy? Let's just keep it as 6 only. 6 means 3 pair. It is easy for me to draw. So basic set will become 3. Right? So this is a basic set, normal condition. 2n is equal to 6. If now beta, if monosomy will occur, condition will become 2n minus 1. Any one of the chromosome from any set will get deleted. So basic set is 3, but deletion is of one chromosome. So is it in multiples of basic set? No. That is why it is aneuploidy type of mutation. Now this is the new condition. Here one of the chromosome gets deleted. So 2n minus 1 is actually monosomy type of aneuploidy. The best example is Turner syndrome, which you see in case of humans, specifically females, when one of their X chromosome gets deleted. So total number of chromosome in a human female having Turner syndrome will become 45. Why? Because one of the X chromosome gets deleted. So Turner syndrome is when human female carries 45 chromosome as one of the X chromosome gets deleted. So 22 autosome pair are okay, but one sex chromosome pair out of which one of the X chromosome gets deleted. Now, what is nullisomy? Nullisomy is basically 2n minus 2 condition. That means, check this. What happened, beta? Here one chromosome got deleted. Now that is monosomy. If the second chromosome of the same pair gets deleted, then that is called as nullisomy. So when both the chromosome of a pair, when both the chromosome of a pair gets deleted, so condition becomes nullisomy 2n minus 2. Monosomy means when one of the chromosome of the pair, when one of the chromosome of pair gets deleted. Tick. So this is known as monosomy, and nullisomy is the condition which I have shown in you the second diagram. Now this is about deletion type of aneuploidy. Next is addition type of aneuploidy under the heading trisomy and tetrasomy. Same thing, 2n plus 1. Here 2n plus 2. So what was the actual condition? Actual condition cell is having total 6 chromosome. That means 3 pair or 2 complete set is having 3 pairs of chromosome represented like this. 2, 3. Now what is 2n plus 1 condition? Check. Trisomy. When any one of the chromosome gets duplicated. So this is the original condition. What will happen, beta? Anywhere one of the chromosome gets duplicated. So this is pair, this is pair. But here there are 3 chromosomes in one set. Sorry, in one pair. This is known as trisomy. Clear? 2n plus 1 condition. So over here, total chromosome will become 2n plus 1, that is 6 plus 1, that is 7 chromosome. This is trisomy. Example is your Down syndrome. Again, it is seen in case of humans. The 21st chromosome pair undergoes trisomy. So the total number of chromosome in humans having Down syndrome will become 47. Normally it is 46, but 21st chromosome will show trisomy. Hence, the total number of chromosome there will become 47. That is 2n plus 1. 46 plus 1. What is tetrasomy? Very simple. See, this was the normal condition. 2n is equal to 6. 3 pair, 2 complete set. Now, what will happen in case of tetrasomy? I hope you all can tell me very easily. Now, tell me what will happen. This is tetrasomy. When one complete pair of any one of the pair of the natural chromosome gets increased. So this is known as 2n plus 2 condition, which means 6 plus 2, 8 chromosome will become in the karyotype. Naturally, it should have been 6, but now here the karyotype changes from 6 to 8 because one pair of homologous chromosome have been duplicated, making total number of chromosome to 8. So I hope what is aneuploidy is also clear to each and every one of you. Moving ahead with the next type of genomatic mutation, that is euploidy. Now euploidy means beta change in the chromosome number but in multiples of basic set. That means if suppose initially cell is having 2n is equal to 6 condition, that means 3 pair. So X will become what? 3. Now this is the normal condition. Now suppose change, maybe either addition or deletion, is taking place with respect to the multiples of basic set. That means if addition of chromosome has to take place, then one complete set has to get increased like this. Right? So number of chromosome will become 2n is equal to 9. Again in multiples of basic set. 3 3s are 9. Correct? This is addition. Now, suppose deletion has to take place, then beta full one complete set has to get deleted. So this is known as deletion type of euploidy mutation. Now here the number of chromosome will become 3. One set is only left. Again in multiples of basic set. 3 into 1 is 3. So whenever change in the chromosome is occurring in multiples of basic set. So what is euploidy? When the change of chromosome is taking place in multiples of basic set. Now there are two types of euploidy, that is haploidy and polyploidy. Haploidy means deletion type of euploidy, where one complete set of the chromosome gets deleted. That means a cell from diploid becoming haploid. So haploidy means when one complete set of chromosome is getting deleted. So this is known as haploidy type of mutation. How? Check this. Suppose this is a cell which is having 2n is equal to 4 condition. 2 pair. Now what will happen if euploidy type of genomatic mutation has to take place? One complete set of chromosome has to get deleted. Not 1 or 2, one complete set. Now that one complete set may have 2 chromosome, 3 chromosome, 4 chromosome, whatever may be the condition. But one complete set of chromosome has to get deleted in case of euploidy mutation. One complete set gets deleted. If this happens, then a diploid cell will become naturally haploid. So this type of euploidy, where a diploid cell becomes haploid due to mutation, is known as haploidy. Now, opposite to it is polyploidy. That means when the number of sets of chromosome gets doubled, or when one complete or more than one complete sets of chromosome are added to the natural condition. So what is polyploidy, beta? When complete sets of chromosomes are added. Complete set of chromosome gets added. That means, like for example, check. If this is a natural condition, 2n is equal to 4. Now, if over here I have to show polyploidy, then what will be the polyploidy? Adding one complete set. See, I am adding one complete set, two complete set. So now the condition will become 2n is equal to 8 in multiples of basic set. The basic set is 2, 8 is also a multiple of basic set. So when one complete or more than one complete sets of chromosome are added, so this condition is known as polyploidy. If the same thing gets deleted, then it is known as haploidy. Whatever is happening, either addition or deletion, it is taking place in multiples of basic set. Therefore, this type of mutation is known as euploidy genomatic mutation. Right? Yes or no? Have you all understood the three types of mutation? Gene, chromosomal, genomatic. In gene mutation, you have seen when the change is occurring in one gene, within one gene. In chromosomal, many genes on the same chromosome. Third, whole chromosome is either getting deleted or added up. Right? Okay. So beta, these are the types of mutation. Now question comes, how mutation takes place? Ma'am, you are being constantly telling that this is mutation, sudden change in the genotype, and these are the types of mutation. But why? Why are these changes taking place? So now we are going to discuss the reason behind mutation. In nature, there are so many agents that can bring change in the genotype that can induce mutation. Such chemicals, such agents are known as mutagen. So what is mutagen, beta? Mutagens are basically the agents which introduces mutation, which causes change in the genotype and hence the phenotype of an organism. So mutagen kya hai? Basically, mutagens are the agents which causes change in the genotype as well as the phenotype of an organism. Right? Now, mutagens can be divided into two as physical mutagens, where you are going to study about the different rays coming from the sun, basically your X-rays, gamma rays, UV rays and all these. These all are potent mutagen which can cause change in your genotype. Apart from the rays, some chemicals are also there which can also change the genotype under the heading chemical mutagen. Right? So how many mutagens we have to study? Two types. Physical and chemical. First, talking about physical mutagens, we can divide physical mutagen into two categories as ionizing. Basically, physical mutagen means radiations. I already told you. So there can be two types of radiations, beta. Ionizing radiations and non-ionizing radiations. Bachcho, ionizing radiations like for example your X-rays, gamma rays. They are very powerful rays which can very easily penetrate in your skin. That's why when your bone breaks, you go for X-ray. X-ray means what? doctor wants to check that where actually your where your bone actually got broke. So he wants such radiation which should penetrate deep inside your skin and take the picture of your bone. So X-rays are very potent enough. They are energetic radiations which can penetrate inside your skin and take a clear picture of your bones, so that doctor can see that, okay, this portion of the bone has got broken, so he mends that portion. Right? Now, even it is said that person should not frequently undergo X-rays. It is not a good habit. Why? Because X-ray are mutagen. If you're constantly in contact with X-rays, if your skin is constantly, if your bones, if your DNA is coming in direct, if is in constant contact with these mutagen, they can cause damage in your DNA. They can cause mutations. How? Because these X-rays and gamma rays can cause the break in the chromosome. Now imagine if the strand, if that chromatin fiber, if that chromosome breaks, then obviously the number of chromosome will change, causing the mutation, causing the change in the genotype and phenotype of an organism. Right? So, ionizing radiations like X-rays and gamma rays, beta, they penetrate the skin and causes breakage in the chromosome of the cell, causes breakage in the chromosome of the cell, thus introducing mutation. Right? Understood? On the other hand are the non-ionizing radiations like UV rays. Because they are not that energetically strong. So, these do not penetrate the skin. ये skin को तो बेटा they do not penetrate. However, they can cause damage in the upper layer. ठीक है, okay, fine. UV rays cannot take the picture of your bone. They cannot penetrate your skin, but at least they can cause damage to your skin cells only. That's why we say na, okay, whenever you go under sun during summers, then always cover your skin or apply all those lotions which are UV protected. Why? Because during summers when sun is at its maximum peak, it is usually giving UV rays along with the other rays, and UV rays, when they come in contact with your skin, they can also cause skin cancer. That is why it is always suggested that if you go in the sun during afternoon time, then try to cover your body or else wear some kind of lotions which are UV protected. Correct? So these are non-penetrating but can cause cancer to skin cells, Right? Now these are about physical mutagen, which are basically the radiations, which are basically the rays. Apart from this, there are certain chemicals also. If you come in contact with such chemicals, then surely they are also going to cause mutation. Chemicals like nitrous acid and base analogues. So first we are going to talk about one of the chemical, that is nitrous acid. Nitrous acid formula is, everybody is a chemistry student, HNO2. Beta, these nitrous acid, they causes oxidative deamination of the nitrogenous bases of DNA. So these nitrous acid beta causes, you just have to remember this line, that's it, causes oxidative deamination of nitrogenous bases in DNA, that causes mismatch base pairing. गलत base pairing हो जाएगी. Adenine should pair with thymine. Then otherwise it starts pairing with cytosine. Guanine should pair with cytosine. Otherwise it starts pairing with thymine. Wrong base pairing means wrong synthesis of DNA. Wrong synthesis of DNA means again mutation. So what is nitrous acid doing? It is causing oxidative deamination of the nitrogenous bases that thus causes change in the base pairing rule. Same thing is done beta by base analogues. So first let me tell you what are base analogues. There are two examples which you have to learn. Bromouracil, 5-bromouracil and 2-aminopurine. Now beta, these are the different chemicals. Base analogues means nitrogenous bases structural similarity is exhibited by these chemicals. Like for example, 2-aminopurine, it is structurally similar to adenine. Suppose this is my DNA having thymine. It is undergoing replication. So if it wants to undergo replication, then in front of thymine, what will come? Adenine will come. This is the only right base pairing rule. Now, suppose the cell is having 2-aminopurine. What will happen? Instead of adenine, thymine will start pairing with this 2-aminopurine because thymine will think that 2-aminopurine is same as adenine as they both are structurally same. So now instead of adenine, thymine will pair with this base analog. Now, okay, thymine has paired with 2-aminopurine. Now, when this DNA strand having 2-aminopurine, if now it will undergo replication, then naturally it is actually replacing adenine. So next DNA strand should also have thymine. But no, it again does the wrong base pairing. It goes and gets paired up with another nitrogenous base like cytosine. So these base analogues, they are structurally representing one or the other nitrogenous bases. They take their position and then changes the base pairing rule, again causing mutation. Right? So what are these? Basically, these base analogues are structurally representing one or the other nitrogenous bases, right, comma, taking their position during replication, taking their position during replication and then later on causing faulty base pairing. And then later causing faulty base pairing, leading to mutation. So basically, these mutagens, these chemical mutagens, how are they introducing mutation? They are simply changing the base pairing rule, be it nitrous acid, be it base analogues, whatever be the chemical agents of mutation, they are simply, the main concept is to change the base pairing rule, cause change in the DNA structure, cause mutation. Understood? So this much is there for the topic called mutation, and for today I'm going to keep it till here. Again, simple, short and crisp. If you revise everything beta, if you have understood so far, then I'm sure you can solve all the questions related to mutations very easily. Before I say bye-bye to everyone, there is a homework for all my dear students. Please try to solve these questions, and apart from these, try to practice more questions which are given to you in the form of your DPPs. Apart from DPPs also, beta, try to solve as many as possible questions, because more the solving power you have, better results you will attain. So thank you for joining me. Bye bye everyone. Take care.